Welcome back. Today we'll talk about the topology of hippocampal spatial map formation. This will help hopefully prepare for 3 talks during next week's IMA workshop. So these 3 talks, one on Tuesday and 2 a week from Wednesday. We'll also discuss, at least at the beginning on Wednesday, some additional stuff related to these 3 talks. All 3 speakers recommended this paper on cell groups reveal structure of stimulus space, so we'll discuss that first. This is the very first paper to use only the spiking activity of place cells to determine the topology using homology. So let's see what that means. We have neurons in our hippocampus. And those neurons can be involved in a number of different things, but we're looking specifically at spatial navigation. So we'll look at how these nerve cells transmit information and how it's related to spatial navigation. So, for example, suppose we've got a mouse entering a maze, and this mouse travels along this maze. When it's traveling along the maze, these neurons are firing, and so let's take a look at neurons firing. So you'll have a cell, it's called a place cell, this neuron, that basically has this electrical pulse type thing that looks somewhat like this. And so in order to interpret the environment, you have a number of these electrical pulses coming along. Maybe we'll have another one. They're not all completely uniform, so something sort of like that, not really. So each of these electrical impulses, they're just going to be represented by a bar. So as the mouse travels along, we've got our cell 1 is going to have these spikes, these electrical impulses, these spikes, and so this thing right here is called a spike train. So cell 1 sends several electrical impulses as the mouse travels along. You can see when it gets to this region, cell 2 now starts firing. And then when it gets to this region, well then cell 3 now starts firing. Notice we can detect the topology of the maze because cell 1 and cell 2, they overlap in terms of their firing. And they know that one of these things that these place cells do, these neurons, the reason they're called place cells is because they fire when you're in a particular place. So cell 1 only fires in this region. Cell 2 only fires in this region, and cell 3 only fires in that region. So depending upon the place where the mouse is, then a particular cell will fire, the neuron will fire, and that's why they're called place cells. And since they do overlap, so the place where cell 1 overlaps can intersect where the place where cell 2 overlaps, we know that this region 1 is connected to this region 2. And so that's why they are called place cells, and that's how basically how we cover the topology of the configuration by looking at the intersection of the spiking data. Any questions so far? So let's take a look at a similar case, but now we've got 2 different environments, one that has a hole in it and one that doesn't. In one case, the mouse might start off with at here, travel along. Let me see if I can get the coloring right. Yes, travel this way, and then go over here. So that might be the trajectory. We can see that if the mouse takes this trajectory, then it starts here in the intersection of the place for 1 and 5, and so thus 1 and 5, that region are both spiking. And then when we're in just this region, we only have spikes only for cell 1. We then enter the intersection between 1 and 2, and so cells 1 and 2 are firing. Then into region 2, where only cell 2 fires. Then into this region right here, between 2 and 4, so 2 and 4 fire. And then into a region where that overlaps cells 2, 3, and 4. And so it then 2, 3, and 4, and then 3 and 4, 4 and 5, etc. And so that's the relationship between these two things right here. This is so C is the environment. This is where the mouse is traveling. So this is the environment in which the mouse or rat, they do this on both mice and rats. And given that, so how the mouse travels in this environment will then determine the spike trains of the different cells. And we can see that if instead the mouse travels in this region here, so you've got one, going along here, we can see that 3 and 4 is next, and then just 4 for a bit, and so this would be another potential path. So a place field, so some definitions, we know what the place cells are. So up here in A, we've got our place cells. So the spiking data for cell 1, the spiking data for cell 2, etc. Down here, we've got our regions in our mouse is traveling in the two-dimensional plane. So this is some subset, of R2. And the place field will be the region in space where the firing rates are significantly above baseline. So this would be the place field for place cell 1. So this is the place field for place cell 1. So this region here, whenever the mouse is in this region, wherever it is, then we will be having that cell 1 will be firing above some baseline stuff. Now, in biology, you rarely have zeros and ones. Usually, you might have some sort of baseline activity and then this spike. And do we count this as a spike? Maybe, maybe not. So things that are significantly above definitely get counted. Things that are just noise, background stuff won't get counted. So we'll just do spikes. So we'll have a spike for this, we'll have a spike for this. And depending upon where you set your threshold, you probably don't have a spike here, but it does depend upon where you set your threshold as to whether or not you've got these spikes. And so there might be some, when the mouse is out here, maybe there is some small, stuff going on with cell 1, but it isn't until we get the spikes that are above threshold that's the region, where the mouse is traveling, that if those things, you have a firing rate significantly above baseline, in that region where the mouse is traveling, that would be the place field for place cell 1. And similarly, you can talk about the place field for place cell 4. And so we've got these different place fields. There is one thing to keep in mind. As in many areas, the same word can be used for multiple things. So a place field can also refer to the function from the subset in R2. So whatever subset you're in here, so some subset in R2, into basically your firing rates. So we could instead talk about the place field that for this point here, we get this particular firing rate. For this point here, we're going to be getting a firing rate for both cell 1 and cell 2. So in terms of the locations, you can also talk about a function from the points where the mouse is traveling into your firing rates. So that's another definition for place field, but that's not the one we're going to use. The one that is mostly used in these papers is the place field is this set, this set where when the mouse is in this region, your spiking activity is above your baseline level. Any questions about that? Well, this picture in the middle is kind of indicative as to what we might want to do. The idea is, can we recover the topology of the space traversed by the mouse by only looking at the spiking activity of the place cells? So in all previous papers, what biologists did was they looked at, they had both this data, they knew where the mouse was when the cells spiked. So they knew exactly where the mouse was when they got this spiking information. So this point of time here might correspond to this point of time here might correspond to that. The biologists initially knew that. And so they knew the path that the mouse took, and it was probably not quite as simple as it probably, meandered a little bit. Mice don't always go in just one direction, but they knew the path. Well, taking a look at this picture, it's pretty obvious what to do if you want to recover the topology. Every single place field can become a vertex. So you can have a vertex between every single place field. And if there is an intersection between the two place fields, we can draw an edge. If three of them intersect, we can fill it in with a triangle. In other words, we are doing our check complex. So just to review, in terms of our check complex, if we do have two balls intersecting, we get an edge. If three balls intersect, we get a triangle, and we get higher dimensional objects as well. Note this is not the RIPS complex, so we're not doing the RIPS complex, where in the RIPS complex we would have filled in whenever you had a triple of edges forming a triangle, we would have filled it in. We're not filling this thing in, and that's to preserve this hole right here. We want to preserve that hole. Now, when it comes to circular balls, if I increase the epsilon a little bit, the hole would fill out. But in one of the papers we'll look at, the regions can be epsilon balls. In this particular, ellipsoidal. They can be ellipses. So we might have ellipses coming along here, and if I've got, three ellipses, then we might have a fairly big hole, and so thus we might want to not fill in the triangle. In this paper, they basically did nice circles, but one can also do ellipsoids depending upon the molecule, depending upon the neuron. So in some neurons, they have circular place fields. Other neurons have ellipses as place fields. So we can get our check complex, and remember the nice thing about our check complex is that this, by the way, is also called the nerve. So our nerve is also similarly just the same thing as our check complex, our non-empty intersections of k plus 1 distinct elements. So we get our nerve. And remember, the nice thing about this nerve right here is that it does preserve the topology. So we do know that if we use the check complex, then our nerve is going to be homotopic to the union of these elements. So we preserve our topology. And that is not true for the RIPS complex. And so that's why we like the check complex. We want to definitely preserve the topology. Any questions about that? So now we come back to this thing here, and I used my place fields to create this, but that's not what we really want to do because the brain doesn't have this information. The brain is trying to determine this information. This is supposed to be the output. So we can have it, but it's the output. This is the input into your brain, and your brain wants to interpret this, so it doesn't have this information. So to interpret the topology, to get this, it needs to come from the input. But you can see that if we do something similar, where now instead the vertices here represent a place cell. So for each of our 5 place cells, we have a vertex. And then we can draw an edge if 2 place cells have spiking within some time period. So if they both have spiking within the same time period, we can draw an edge. If 3 of them have spiking within this time interval, then we can draw a triangle, and so we can get the simplicial complex that way instead of this way. So we don't have this information right here, so we need to get it from the input, the spike train data, and then we can determine the topology. So the question is, how can the brain determine the topology of the space traversed by the mouse by only looking at the spiking activity? And so that was one of the main contributions of this paper, that they didn't use the output information to gain the information. They just used what the brain has, and that is the spike train data. Now there is others, so I am giving a simplified view. You can talk about the magnitude, the width of your spike, so there's a bunch of additional information, but right now we're only looking at the spike train as our input and ignoring any other potential piece of information. And this is a nice on and off type thing for determining topology. So our vertices are our place cells, and then we add a simplex if the place cells co-fire within a specified time period. In other words, what we're going to define is cell groups. So cell groups will be a collection of place cells that co-fire within a specified time period, and they do have to co-fire above a specified threshold. If I just see, one thing here, maybe that's not enough above the threshold, so that might be ignored. So there is always noise in your data. You want to be able to handle noise. So as long as they co-fire above a specified threshold, then we will put all those cells into one group. So this right here, cell 1, is its own group because it does co-fire with itself. Cells 1 and 5 are also a cell group. Cells 2, 3, and 4 are also cell group because they also co-fire within a specified time period. And then our simplices will correspond to our cell groups. And so if we've got 3 elements in our cell group, 3 cells that co-fire within the same time period, then we get a 2-dimensional triangle. So the dimension will just be the number of place cells in our cell group minus 1. We get this nice 2-dimensional cycle. So that's the idea, that we go from the spike train data, that's the brain input, then we can create this simplicial complex, which should correspond to the topology of the actual environment. So this is a way for the brain to interpret the environment, is to actually do this, create a simplicial complex. Your brain can create a simplicial complex. At least that's one way for us to understand the brain input and how it corresponds to interpreting the environment. Any questions up to here? Because this is the main idea. So the idea, again, is by saying that we can recover the topology of the space traversed by the mouse by looking only at the spiking activity of place cells. Unfortunately, at the time when this paper was published, the data, the biological data, did not do full coverage. If I want to detect a hole, if I've got this hole right here, if my mouse only travels in this region, I'm not going to detect the hole. So we need to have sufficient coverage of the region in order to detect the topology of the region, and there just wasn't enough biology data at that time. So instead, their paper was a proof of concept. Their data was obtained via computer simulations of mouse trajectories using biologically relevant parameters. When I was a graduate student and I was first listening to talks, I would think, oh, this was a great result. And then I was thinking, wait a minute, was this simulated data or was this experimental data? One always had to pay attention to whether or not one was simulating the data or one was, experimental data. They both have very important, roles. If you understand the data, that means you can simulate it. If you can't simulate the data, then you probably don't fully understand it. So simulations are quite important. In this case, they did the simulation to basically, prove their idea that if we take a look at these mouse trajectories, and so in this particular case, we have a mouse trajectory. I can't quite see where this thing is going, but these green lines is basically where the mouse is moving along here. I've just sort of made that up, but they did a number of trajectories and then looked at creating the place cells. So let's go ahead and take a look at what the computer simulation did, but it was biologically relevant parameters. So in other words, if they could not detect the topology using this simulated data, then there was no hope that their idea worked. But if they can, and they did detect the topology, well, since they used biologically relevant parameters, there's a good chance it should work with experimental data as well. And so they generated basically this random walks. So they generated random walks. So in one case, they had the mouse traveling within a square. And the sides of the square were L. L was frequently taken to be 1 meter. And they know basically what the speed of the mouse was if, L is 1 meter. And so you had a mouse traveling along here in a square. But you also could have had the mouse traveling along where there's a hole. And so maybe this region is inaccessible to the mouse, and so it might be traveling along here with this hole. Or maybe there's 2 holes, 3 holes, or 4 holes. So they basically did a random walk in these 5 different environments, and the total duration in order to cover the entire space, they only needed about 50 minutes worth of data. So the green is basically the path that it's traveling, and so traveling for 50 minutes, so can basically recover this thing here. All the circles is the place fields that they then generated, which we'll talk about shortly. Any questions here? So we first start off with generating random walks. They did 300 trials, so 300 trials, and they placed 70 place fields. And so they basically just randomly chose points. So they randomly would choose a point and then place a circle around this point, and the circle, the radius of the circle would be 0.1 to 0.15 L, where remember L was the side length of the square. So the centers were initially just chosen uniformly, so we do some kind of distribution so that once I have covered these 3 regions, then we take a look at the complementary regions, the remaining part, and then each point within this remaining part, there's an equal probability that we'll choose this point and then create a place field at that point. And then once everything was covered, then remaining points were chosen uniformly at random. So then once everything was covered, we can just throw a dart, and then wherever our dart landed, we then choose a place field with a radius somewhere in between there. Again, just chosen uniformly at random. So that is basically how the data was generated. Any questions there? Well, we now have, so we have the environment. The environment was this box with the mouse traversing it. And then we knew that a place field here corresponds to a spike train. So if we call this cell I, so if this right here is cell I, place field. If this is place field, the region within the rectangle or the rectangle with holes, the square with holes, then we want to go out and get the place cell and get the spike train for that. So first they generated the environment, the random trajectory, then they generated, so first they generated the random trajectory of the mouse, then ignoring the random trajectory, they also generated the place fields by basically randomly taking points and radii. And then after you've got a place field, you need to correspond that to a place cell, and so to create a spike train. They know from biological experiments that basically spike trains follow this inhomogeneous Poisson process with constant rate, and so as you pass through this place field, so as the mouse passes through this place field, they know from biological experiments what a spike train should look like as you pass through, and so you can just, using a Poisson distribution, go ahead and generate this spike train. So that pretty much finishes up how they created it, but then we're going to talk a little bit more about their assumptions in terms of their model. Questions up through here? The next thing, however, is that data always has noise. So they then added noise. So to add noise, they basically deleted spike trains. So R% where R was someplace between 10 and 1%. So between 10% and 1%. And so they might have just gone ahead and deleted, say, we might delete this spike here. So we choose a spike at random, delete it, and then move it elsewhere. So this might cover, this might cover several time intervals. So when we're, going through this sort of thing, you might have, this spike train right here covering several time intervals. Maybe we will delete this one and then move it over here. So, so it's added back irrespective of the position. So maybe I will delete this one and we'll add it way over here. So noise was added by deleting spikes and then just adding it back at random times, ignoring the position along the trajectory. So if you had a bunch of spikes here and nothing here, well, we might delete this one and add it way over there, and that gives us our noise. Any questions there? Well, whenever one does experiments, even if they're computer simulations, controls are very good. They are required. One should always do controls. What if we basically just had random data? Would we be able to recreate the topology with random data? Well, we hope not. But basically, they shuffled the data. So the data sets, remember there were 5 different environments. We had 5 different environments, one with 0 holes, one with 1 hole, one with 2 holes, up to one with 4 holes. And so you had this spike train data. Maybe this was the data for 0 holes. And then maybe we had this other data when we had 4 holes. And what we can do is we can just put this thing into the 0-holed one, put this thing into the 5-holed one, or sorry, the 4-holed one. There's no reason to do that except that you want to create a data set that doesn't come from any particular environment. So if we mix up the environments, if we shuffle the environments so that our data doesn't come strictly from the 4-hole data or strictly from the no-hole data, but if it's shuffled together and pooled them so that we don't create a single environment, then we shouldn't be detecting a particular environment. So we should be having problems with our data. So they did take a look at this control as well. Make sure you always have controls if you're doing experimental or simulated data. Questions? So they made several assumptions about their place fields. One of the things that they assumed is that it was omnidirectional. So what that means is it did not depend upon the direction that the mouse traversed the place field. So here I've got no cell firing of this. So if this, say, corresponds to cell I, if this is the place region corresponding to cell I doesn't fire. Now cell I starts to fire, and then it doesn't fire anymore. Well, I might travel this way, or maybe we'll travel this way. We're assuming that the place fields are omnidirectional, so when we go to the place region, the place field, and output the spike train, it will not matter the direction. So the Poisson process will not be affected. The way we generate the spike train will not be affected by how you traverse, the direction in which you traverse through this place field. Any questions there? Well, in reality, this may or may not be a correct assumption. It does seem to be a correct assumption in an open field. So in an open field, it doesn't seem to matter which direction you travel. It doesn't seem to matter at all which direction. But if there is a linear track, place field cells can also, these neurons can also detect angles and directions. So place cells, these neurons can detect more than just location. Right now, we're just saying that the spike train depends only on our location. So the spike train should only depend upon where you are and not whether you're heading up or whether you're heading down. But some place cells can also determine the angles. So depending upon where the mouse is and what cells you're looking at and what animal you're looking at, you might also get angles and direction as well. And so if I were to do that, then when you're taking a look at your place cell, going up will be different than coming down. And so the topology corresponding to a nice linear track will actually, you go up, you come down, and you actually create a circle. So, so one can create a circle in order to actually, so that would be the topology corresponding to a linear track. But we're working in open fields, and with open fields, this does seem to be a correct assumption that the direction doesn't, you can ignore the direction in terms of analyzing these spike trains. Questions on that? Assumption 2 is that place fields have been previously formed and are stable. So basically, what this means is that if you put the rat into this maze, you will get a particular place field, and it will form within the brain. So cell 1 will represent this, cell 2 will represent that, etc. But if we then take the mouse out of this region and put it into a region where there's this hole, maybe there is a tree in the middle, so it can't go into this middle, but whatever causes the hole, there's a hole that the mouse cannot go. And so the mouse cannot go into this region at all. It's forbidden. Maybe there's a tree and you can't walk through trees, so you can only traverse in here, the place fields will actually look different in the two regions. And so we will assume that the place fields have been previously formed and that they are stable. For periods of time they are, but actually if you, depending upon the experimentalist or the experiment, how stable it is, whether it lasts for minutes, hours, weeks, it will vary. These are all experiments where you've implanted something into the mouse brain and maybe that's moved a little bit. Around the hippocampus region. But it can actually undergo very rapid context-dependent remapping. So here is an example of just in terms of put the mouse in a circular environment or in a square environment. These are both open fields, just one circular and one square. And so in this 2002 Nature paper by Lever et al., they looked at the stability of place fields, and this was actually after 21 days. But you can see that after 21 days, cell 1 in the circular field corresponds to this region. And you can see there's actually a denser part, a less dense part. But this is the place field for cell 1 in the circular environment. This is the place field for cell 1 in the square environment. And you can see that the regions can vary a bit in terms of, and you can actually get more than one region as well. We're going to ignore the more than one region probably until Wednesday, but we will talk about there are actually place fields that can be disconnected. There aren't many of them, probably less than 10%, but you can actually have place fields that are disconnected. And so in this case, in the round environment, it's disconnected. In the square environment, it's not. But you can see that the location of the place field will vary depending upon the mapping. And so if you take the mouse out of this thing and put it into the square environment, its brain changes. So it might be walking along, it might be walking along like this in both environments. So maybe, actually, I think probably mice don't do this. I think mice, might do more like that, but it might do the same path. I don't think mice are good at repeating exactly the same path, but, suppose it did. Whether it did the path in this thing or whether it did the path in here, where place cell I fires is different. And so you can take the mouse out of this and put it into here, and now we've got the mouse will very quickly remap. And so what place cell I means now, it will have a completely different region. And then you put it back into the circular one, and then place cell I, what it means again, the place region, will again change. And so you can have this remapping very rapidly. So we will assume for this paper that they are stable and that they're, for this fixed environment and that they have been formed. But there is a time in which it does form. And we'll actually talk about that on Wednesday using persistent homology. So the next paper we'll take a look at, we'll actually use persistent homology to actually talk about how the place fields form, and so how this remapping is done when you change the environment, and how a mapping is first formed between your cell firing and your place regions. Any questions about that? The third assumption is that the collection of place cells covers the entire traversed region. So again, if I want to detect a hole, if the mouse just travels, along here, and that's all it does, I'm not going to detect the hole. We have to come here and join these things. We have to traverse this region as well. And so the trajectory must be dense enough to sample the majority of cell groups. 50 minutes seem to be sufficient to do that. depending upon the number of cell groups. There are actually millions of cells that are involved, but in terms of experiments, you can't detect millions of cells. Maybe you can detect 50 cells, maybe 100 cells, maybe. And so in their simulated data, they only looked at somewhere between 70 and 140 cells. But that is sufficient to actually detect the topology as long as the mouse traveled. So we need to have our path that is sufficiently dense. Our path needs to be sufficiently dense within this region to cover it. And as I said at the time of the paper, there wasn't a sufficient density biologically, so that's why they simulated the data. Biologically, place cells do cover the environment multiple times. So in terms of the place cell coverage, so you also want to have, this is the collection of place fields, not the path. So in this case, we just want to make sure we have sufficient coverage. So ignore the green path. That's a later assumption. So in a later assumption, we'll talk about the green path needs to cover. But if if these are my place cells and for some reason I don't cover this region here with place cells, we have to have place cells that will correspond to this region here. Otherwise, you don't get spike train data corresponding to this region. And so you do have to have sufficient place fields to cover the entire region. And between 70 to 140 were sufficient, even though we have many, many more place cells, orders of magnitude more in our actual brain. So our place cells multiply cover the environment. Biologically speaking, and simulation, they also need to cover. Basically, double coverage is quite sufficient. Don't even quite need double coverage, but double coverage will give you the correct topology. Questions there? The other thing that they did is when you're trying to compare the homology, I want to see that there's a hole in this data. So I want to see that there is this hole right here. To detect this hole, well, I only need to know H1. But if I actually want to recover the topology, I'd like to know the number of connected components. This has only one connected component. So I'd also like to know H0 to make sure I don't have two connected components. I want one connected component. Then I want H1 to detect my hole. But what if in this data we generated something like this? If we generated a sphere like this that is open in the middle. Suppose we had non-trivial H2. Well, this mouse is traveling in the plane, so we should not have non-trivial H2. Now, we might have multiple things intersecting, so we could get more cells in here, and so we can get higher dimensional simplices. If 4 things intersect, we can get a solid 3-dimensional tetrahedron, but topologically speaking, I can flatten down, this to just be a triangle which will have the same homology. So it'll be a different topology because this was 3-dimensional and this is 2-dimensional, but it'll have the same homology, and we'd like it to have the same homology. We don't want any of these 2-dimensional holes in it. We don't want any higher dimensional holes. We just want a 1-dimensional hole. And so they actually calculated HN. They calculated HN for n equal to 0, 1, 2, 3, 4, and they stopped there. So they calculated HN up to n equals to 4. And if you want to calculate, accurate homology, then you do need to be able to detect the n+1-fold intersection. So we do need to be able to fill in. So if I have this tetrahedron right here, so if I've got this tetrahedron right here, I might have these edges right here that intersect, but I need more than that. I need them to intersect completely, so maybe I want to add another, I want to fill these things in So I want to make sure that I've got sufficient coverage so that all 4 of them intersect, so that I get a solid tetrahedron with all 4 of them intersecting, so that I'll get the correct homology. We will also need the path to go through here, because if I have a path, so if I've got a path that comes through and my path, say, traverses around here, then if I never enter this region, I'll think this is a hole as well. So I also need the path to go through here so that I get the spike train data that will correspond to the intersection of 3 of them. So 3 place cells firing within the same interval. Questions up through there? Okay. The other thing that's pretty obvious is we need holes to be larger than the diameter of the place field, because after all, if I've got this large place field, my place fields are not this large, but if it were, it would cover up this hole and I would never detect it. And so we do have to assume that our holes are larger than the diameters of the place fields. The other sort of things is each connected component will be considered convex. The main thing is we need convexity of the intersections. Remember, we want the nerve lemma to hold, and as long as the intersection is nice and convex, then it will work. The nerve lemma will hold, and we will be able, the homology will be the same for both the simplicial complex as well as for the union of the place regions, of the place fields. We also can't have too much noise. If there's too much noise, then our spike cells won't mean anything. And we will do roughly circular with similar size, although they did check to see if we did do different sizes. So if we had a place field that's smaller, a place field that's larger, and allowed a variety of sizes, they did check that out as well. And depending upon the change in size, they were still able to reconstruct the topology. Just one comment here is we mentioned multi-peaked. Remember, we might have a place cell that might activate in this region, but it also might activate in more than one region. That is multi-peaked, but we will ignore that today and just talk about single peak. So each place field will correspond to a single place cell and vice versa, one-to-one correspondence. But we'll, on Wednesday, talk about multi-peaked. Okay. So, again, our data with those assumptions is now just the spike train data. So now we're looking at just the spike train data. Yes, this data was generated by doing random trajectories, using randomly placed place fields. So using randomly placed place fields, we outputted our spike train, but now we have our spike train data, and we now want to look only at our spike train data. And so now we're back to this picture here. We now have our spike train data. We can now create our simplicial complex. We would expect that that would recover our topology, and it does. But remember, we don't have an exact one-to-one correspondence between our spike train and our place fields. Yes, we did go and generate our spike train data from our place fields, but we used a Poisson distribution, so that's, one little, variability that we can have. And the other thing is we added noise. So this is noisy as well. So with the added noise, the question is, can you still generate the correct topology that will correspond to your environment? And the answer is, they calculated the homology using GAP software. So if you want to write your own programs, you might consider using some of the packages within GAP. And so this is basically the recovering the correct topology. The definition of correct is I want HI to be correct for 0, 1, 2, 3, and 4. In other words, in this case, H1 equals to 1 because we've got one hole. in this case H1 equals to 0, we have no homology for 2, 3, and 4, since it's supposed to be, two-dimensional, and it's connected, so we've got H0, we've got one component. And so as long as all of these are correct, so 0 for anything higher, 1 for H0, and the number of holes should give us our H1. So we can see that it doesn't seem to matter whether we've got 1 hole, 2 holes, 3 holes, 4 holes, or no holes. It's pretty equally as good. What it does affect is the noise level. So as we increase the noise level, the percent correct does go down. But you can see even at 5%, we're getting pretty good correlation of the topology. So the percent correct, over 90% is correct even when you've got 5% noise. And even here, we detect the topology most of the time. We will finish up this paper talking a little bit about geometry next, as well as the persistent homology of another paper. I'll post those links this afternoon to both this paper and the paper we'll discuss on Wednesday. Any questions? Okay. So see you Wednesday.